The overall goal of this histological and imaging protocol is to present a robust system that preserves the epitope antigenicity and structural integrity of skin, which allows for full exploitation of confocal imaging modalities.
This method can help answer key questions where there is a requirement to examine the nature of any type of cell within a complex three-dimensional tissue architecture. The main advantage of this technique compared to standard histological techniques is that it is robust and easy to perform. Additionally, the protocol perfectly complements three-dimensional analysis in confocal microscopy.
Begin by trimming skin harvested from the dorsomedial region of a mouse into rectangular pieces. Each piece should be sized to fit in the bottom of a cryomold. A 1 square centimeter area of dorsal skin, which fits into a 22 mm by 30 mm by 20 mm cryomold, is demonstrated here. After fixing, push the washed skin to the bottom of the OCT-filled mold so that it lies flush with the bottom. Mark the hair follicle orientation on the cryomold, as this determines the orientation of the cryostat cut. Transfer the cryoblocks onto a metal plate in the -80-degree freezer to prevent floating and dislocation of the tissue. Monitor the freezing process to maintain the orientation of the skin at the bottom of the cryomold, since unseen air bubbles can cause the skin to rise.